Thanks for the introduction, Corey, and thank you to the organizers for allowing me to speak today. Really excited to share some of the work we've been doing in technology development for resolution of heavy chains and how we're applying that technology to questions in autoimmunity. No. I'm not. All right. So it doesn't come as any surprise to people in this room, but antibodies are really important for a variety of different immune functions. And Corey set me up for this pretty well. So the variable region has been well characterized, and it's mainly focused on antigen specificity. So a lot of the allelic discovery has been focused on this locus, but for the constant domain, it's really largely misunderstood in terms of the breadth of diversity that's there. This can have a wide range of effects on some of these effector functions that we know that the constant region dictates, such as complement activation, antibody-dependent cell cytotoxicity, and more. So current AIR methods are really limited in terms of their resolution. So many of these strategies only capture VDJ gene segments. VDJ race is a common practice used in the field, and it captures partial CH1 resolution for isotype determination. And really the limitation of these methods is revolving around next generation sequencing. So with these transcripts being around 1,500 to 2,000 base pairs in terms of IgG and IgM, next generation sequencing is not able to sequence that long in terms of the length that needs to be resolved in the heavy chain. So what we've sort of tackled in the field is the development of FLAIRR-seq. This allows for near full-length resolution of the heavy chains. So this revolves around the usage of 5' race PCR. We amplify cDNA and then come in with a targeted barcode to either IgG and IgM in this case. Then we sequence those libraries on the Revio by Pacific Biosciences. The average read quality comes out to about Q60, so really highly accurate reads. And just to compare this methodology with that of next generation sequencing, we're able to resolve the full VDJ cassette as well as get subisotype information and allele resolved data as well. So for a methods publication, we wanted to put this technology to the test. So we had 10 healthy donors where we had PBMC from them, and we performed IGH capture, which William Lees introduced, where we generate hi-fi reads on the sequel2E in this case when this was published. We map them to the T2T reference, group them by haplotype, and assemble their haplotype-specific reads for a ground truth genotype. We then took FLAIRR-seq and we wanted to see how well it could do in terms of calling these alleles back to our ground truth. We performed a similar methodology, but clustered those reads at the resolved alleles and compared them back to the iGenotyper output. And what we saw from this exercise is that there's an extensive amount of constant region diversity. So this tile plot is very, very colorful. I hope you can appreciate it. But on the y-axis is going to be each donor ID. On the top there is and the x-axis is going to be each gene. And the color that fills in the box is going to be representative of the haplotype. So you have different alleles represented by the colors. And from this exercise, 28 out of 32 alleles that we resolved from this data set, and just 10 individuals were completely novel or novel extended in the data set. And a lot of these alleles were able to be recapitulated in the FLAIRR-seq data, and we even picked up evidence of IGHC duplications, which are denoted by the star in the columns. If you have 3 boxes instead of 2, we were able to pick up on a duplicated gene. Being quite slow. So really where this comes in, using these two technologies together, is once we have the ground truth genotypes, we can layer on FLAIRR-seq data and look at repertoire expression at the subisotype allele resolved level. So with current technologies in NGS, you're usually only able to get subisotype resolution to some degree, but with FLAIRR-seq you're able to get the expression at the allele level. And also we can make Specific analyses looking at differences in gene usage across the molecule, where if you can have a scenario if you're looking at a certain constant region gene that you're interested in and how its V gene usage is correlated to that allele. So the chords on the Circos plot here really represent the sequence frequency that constant region allele on the top of the Circos and the gene usage is on the bottom in terms of V genes. What we're really able to do with this technology is to apply it to disease settings where we know subisotypes are very important. So we wanted to apply this technology to myasthenia gravis. So there's two main types of the disease. The one that we're going to focus on for the work is acetylcholine receptor antibody positive myasthenia. And this disease is shown to be driven by IgG1 and IgG3 autoantibodies. And this disease normally causes weakness of muscles and disruption of muscle communication. Usually presents first with ocular weakness and can progress to more voluntary muscles as well. So the disease can affect people at any age, but it's more common in women younger than 40 and men older than 60. And there's no really known treatment for the disease. Most of the therapeutics are palliative at this point. But for AChR MG, the pathologies have been well demonstrated with patient sera that these autoantibodies are acting in three manners. So the first is complement activation, where these autoantibodies are binding the AChR receptor, depositing MAC on the neuromuscular junction membrane, and leading to destruction of the architecture. You can also have a modulization of the receptor and blocking of the acetylcholine receptor as sort of a functional block. So we proposed for a grant for the Myasthenia Gravis Rare Disease Network, and were awarded funding to profile repertoires and genotypes from 48 diseased individuals and 48 healthy donors that are all ethnicity and race and sex matched. We're then able to use iGenotyper and IG capture to provide ground truth genotypes for these individuals, and also profile which genes are used in the repertoire by using the FLAIRR-seq technology. So it's been shown in other studies that there are certain variable genes that are upregulated in myasthenia, and we wanted to see if this is recapitulated in a subset of our completed FLAIRR-seq resolved repertoires. The first one of these V genes is going to be IGHV169, which was shown to be upregulated in our data as well. And this is IGHV434 is another signature that we were able to pick up on. But where FLAIRR-seq really shines is we're able to break down this usage at the subisotype level. So it's going to be really important to see in terms of these V gene or variable region genes if they associate with certain constant region alleles or genes used at a higher rate. We were also able to perform, that's supposed to be part. Okay. But we were also able to perform preliminary genotyping in terms of using our capture data. So what you would see here is that 159 unique alleles were resolved, and 137 of those were completely novel. So really 86% of the alleles that we were able to capture haven't been described before in the current database. We're also recapitulating these alleles in another study by Uta Lakiana and Corey Watson's lab. So he's looking at a set of 1,000 genomes project data set. So we're seeing those same novel alleles pop up in this data set as well. And interestingly, 19 of these novel alleles are found in IGHG3, which is known to be one of the autoreactive subisotypes in the disease. So it's going to be important to get those alleles characterized to see if they could have some sort of differential function. So really where we're going with this is annotating a lot of these variants. So right here is a multiple sequence alignment of mostly IMGT resolved genes or alleles. And at the bottom is ones that we've characterized as well. So it's been picked up on in the field that there's IGHG3 hinge length variation. And this is associated in a study recently published with COVID-19 disease and severity. But importantly, these hinge variants resembling the natural 2 and 3 exon allotypes, so with the hinge deletion that I've highlighted here in red, are known to enhance complement activation and complement-mediated cell lysis. So since one of the pathologies that's associated with MG in terms of autoreactive antibodies is complement, we were highly interested in looking at this structural variation. So I've sort of diagrammed this here, where your major allele or M allele has the 4 hinge-linked exons. The S allele has 1 hinge deleted, and then one that we characterized in the data set thus far only has 1 hinge. So we compared these genotypes between healthy donors and myasthenia gravis diseased individuals, and we saw that this structural variation representing the hinge deletion was far more prevalent in MG individuals. It was really interesting that this actual individual, we had one patient that exhibited this so far, where they had, in the reference, you would have 1 copy of each gene, but in the structural variation in this individual, they had a duplication of IgG3 and also IgA as well. So it's going to be really important moving forward to characterize not only these structural variants, but also residue differences between these 2 groups to see if these constant region alleles are having a differential effector function. So this is not only able to be seen at the DNA level, but we're actually able to resolve this in the FLAIRR-seq. So this is just an IGV screenshot where you have the DNA capture contigs at the top showing the 2 haplotypes, and you're able to recapitulate this data in the FLAIRR-seq looking at the RNA where you have the presence of that hinge link deletion showing up in the other haplotype as well. So it is making it into the RNA, and we have sent these sequences off to our collaborators to then test for differential complement activation. So I don't really have time to go through the conclusions, but I hope I picked up on the major talking points, and I'd like to thank sort of all the collaborators that I have and my mentors, Melissa Smith and Corey Watson, for their support. Thank you. Yes, I think this gets to what William Lees was touching on a bit. So in the publication for the FLAIRR-seq method, we had to come up with a way of classifying things as completely novel and uncharacterized from IMGT, or an extension of what was already in the database. So if it's novel extended, that means it had a name in the IMGT database, but we were actually able to fill in the rest of the sequence. So it was sort of a substring, if you will, of that allele. Thanks. Oh. Yes, we're able to recapitulate. Yeah, we're able to see the expression of those duplicated alleles in the RNA data as well. Oh, okay. Is the recombination of heavy and constant genes expected to be linked at all? That's something we haven't really looked at yet, but something that we're eager to look at. If there's any bias between, say, a certain allele that always pairs with a B gene allele, that's a really high interest. If there's some sort of exclusion that certain subisotypes can't express a certain VDJ, it's more biased towards the other haplotype.
Anybody else typing?
Oh. Is a SNP sufficient to find a novel allele? What is the minimum difference required? Yes, a SNP is able to classify a novel allele. Yeah, I think the functional interpretation from the constant region variability, that's a really outstanding question, one that we really want to tackle in terms of testing these interesting variants that we see, because it doesn't only come down to the hinge length polymorphism I showed that we want to test. There's also evidence of where you do have a SNP creating a novel allele that may create another glycosylation site, or you have a residue that's been characterized in the literature before that may cause differential complement activation, things of that nature. So it's really unknown and things that we want to really get at.
Okay. Last, but certainly not least, is Chaim Schramm, who is chief. He's a chief. Yeah, that sounds cool. He's chief of IBIS core at NIH Vaccine Research Center. Chaim is a longtime AIRR-C member, and I'm glad that he's finally using long-read sequencing. And so I'm excited to see what these crazy monkeys look like.
I promise the title only sounds one, but anyway. So just before I start, I want to acknowledge the two people who've done most of the work on this project. Simona Lubo is a postback in the group, and she's really driven this project from basically nothing 2 years ago to starting to get some really nice data that you can see today. And of course, William Gibson, who's a postdoc with us, who's in the group, in the audience here, and he's helping us develop our own immunogenetics expertise and really get this done solid. So just a quick background on methods to not take up too much time. We're working here with Mauritian cynomolgus macaques. These animals were geographically isolated and had a founder effect about 500 years ago. And the thought, which we'll get back to, is that this group of monkeys would have limited genetic diversity and make for a more tractable starting point. So you've heard many times this afternoon from William, Lise, and from Jana just now about how crazy the IGH locus can get. And the goal setting out here was to use these animals to make it more tractable. And you'll have to judge for yourself how well that worked out for us. We did whole genome sequencing, not we, but our collaborators at University of Wisconsin did whole genome sequencing using a combination of nanopore and HiFi. These were assembled with HiFi ASM, and the IGH locus was identified by BLAST and extracted for annotation. So just to show you an example of how we're getting phased assemblies. So this is one particular animal with the coverage across the whole IGH locus and some zoom in just to show you that we're getting pretty good phasing and resolution. We identified initially about 10 haplotypes. These are still a little bit provisional as we go through them and do QC. You can see that 4 of the 13 animals are actually homozygous, so we do have some bonus there, including at the bottom here, macaque 973 is also homozygous in the MHC locus for a relatively rare haplotype M6. And so we have some ability to go to that animal's progeny and do some additional sequencing and really try to understand the population structure of these haplotypes. One of the interesting things we can do with the haplotypes that we have is to start to look for the development of the locus over time so we can see some complex crossover events. So this is using haplotype H1 as a reference and a dGNe's dot plot with some of the other haplotypes that we've found labeled on the side there. And so you can see that there's a block of this, just to point out these are cut off at 90% identity. So anything less than 90% identity doesn't show up here. So you can see a block of high similarity there, the 5' end for H1, H3, and H8. Then you have a different block here that's present in both H4 and H10. Another block that's present in H1, H3, and H8. Another block down here, and then a place where basically everything's completely different. And show this schematically, so just from these 5 haplotypes, you can see these complex crossover and recombination events. And so it's going to take us a while to sort out exactly what's going on here. But we have the advantage of having people with the expertise in the macaque immunogenetics on the team, and also, or specifically the MCM immunogenetics, I would say, and the ability to maybe go back to some more MCM in the colony and try to work out the population structure. It also gives us the ability to start doing comparative immunogenomics. So this is just the most common haplotype that we found, H3, aligned against REMAC10. And so you can see that, actually reasonably good alignment over most of the locus, but clearly it's a different species and there is definitely variation there. But then we took these haplotypes that we had identified and we annotated them using an in-house tool we built called Alligator. I won't spend any time on this now because I'm giving a lightning demo on this tomorrow afternoon, so you can come to that if you want. Or you can find the poster on Thursday and I'll tell you about it there. So just what do we find? So looking at the allelic content of these haplotypes. So on the left is a histogram of just identical coding sequences and how many monkeys they are identified in. So of course, many alleles only show up in a couple of these monkeys because the haplotypes are so different. But H3 is the H3 haplotype is present in I think 7 or 8 of the monkeys, and some of the haplotypes have contiguous blocks as we just showed you. So overall, there's 349 unique alleles identified in these 13 monkeys. Of those, actually a fair number, 265, about 70%, are previously annotated in the gold standard KIMDB from Karolinska Institute. So that's actually pretty good representative coverage, but we do see 30% of these alleles are still not documented. In terms of validating some of these novel alleles, we can show read pileups using the ONT or the PacBio to cover entire alleles, coding of alleles, so to show that we're not getting artifacts there. These look pretty strong. And then you've heard a lot already this afternoon about what defines a gene and how to make a gene. And given that we're still having trouble sorting out, the haplotypes, we're not ready to assign gene nomenclature. So we've used Piglet, which William Lee's introduced before. And so when we use Piglet with the default settings, we get 129, I believe it is, ASCs, or allele similarity clusters at the default 5% cutoff. So this is just the big circle is just showing the whole violet dendrogram from Piglet, and a little zoom in there so you can see those white boxes with the little black arrows there. You can see in the zoom in, those are alleles that are exactly the same, or same up to the nucleotide 318 cutoff. And so because we do have some that appear in multiple monkeys, or some that appear in both our monkeys and KIMDB. When we try to do an alignment of all the alleles that we find in the locus, we get something awful like this. So this is just a tile plot, but it's each box along the x-axis is a separate ASC. So I showed you there are 129 ASCs, and each haplotype has on average about 70 alleles, but this alignment is 275 positions long. And the alignment's not great. When I look at it, I can see just manual things I could do to correct it a little bit, but only a little bit. Like basically this is, and so this is again with the default cutoff for ASCs. So maybe if we adjust the cutoff, use a 6 or 7 or 8% cutoff to define an ASC instead of 5%, this might clean up a little bit. So but you can see here that the allelic content of these haplotypes is not at least contiguously aligned. In summary, so we do here have the first high quality reference assemblies of cyno IGH, or at least what we might call reference assemblies, although we just heard from Jana about how iffy that is as a concept. This allows us to start doing comparative immunogenomics between cynos with rhesus and humans. We can start looking at how the locus changes over time. So again, these are animals that as of 100 generations ago, there were probably only 8 to 10 haplotypes present. And so we can use that founder effect to really look at these recombination events and how the locus has changed over time. And we did whole genome sequencing on these animals, so we're still working on IgK and IgL, and TR alpha and TR beta are in the pipeline as well. With that, I'll just acknowledge the whole team. Danny Duke is the chief of the human immunology section, so he's our PI. I run the IBIS core underneath him with Chris, William, Simone, Koki, and other William. And of course, our collaborators at UW, Madison, David O'Connor, Roger Wiseman, Trent Paul. Take questions. What is the relatedness of these crazy monkeys? So they're all Mauritian cynomolgus macaques, right? So they're all limited genetic diversity, derived from a strong founder effect 500 years ago. In terms of recent relatedness, it's hard. We only have records sort of within the breeding colony, but we don't have great going on. So we can speculate that 973 is probably a product of a consanguous mating because of being homozygous for some of these rare haplotypes. But But beyond the breeding records of the colony, we can't really say too much. What is the criteria for defining a new allele? So for alleles, I've just gone by strict nucleotide sequence in the coding region. So it's one nucleotide different. We define a new allele. Although we're also, even though we are extracting the leader exon and the intron and stuff like that, we're not looking at that when we're defining alleles. So you could imagine defining alleles even more broadly than that. But then for genes, though, or what we're using in place of genes are these ASCs from Piglet. And right now we're using the default 5% cutoff, but I think that we'll probably adjust that. What are cynos used for? They're like rhesus macaques. They're an experimental model used in human medicine. So vaccines, drug trials, all the same things rhesus are. Have you validated these new alleles? So I showed you one particular example. We validated a few of them. If you mean by like direct sequencing, no, but I'm not sure that's particularly necessary given the data that we have. Do these monkeys have GDTCR? Yes. So the delta TCR is in the alpha locus, and so we definitely have that. We have not done gamma TCR, but since it's whole genome sequencing, we presumably could do that. We just got to work our way down the list. Okay, I think that's everything. Thank you.